Hello everyone, my name is Monet Porter and today I'm going to be presenting on synaptic plasticity. Now in order to understand what synaptic plasticity is, we have to define it. So synaptic plasticity is basically the ability for synapses to strengthen or weaken over time. And this is done by the brain being able to rewire itself when new information is present. So typically, people believe that people with the most synaptic plasticity are children, which is why they tell you to teach your um, newborn babies as much information between like the ages of 0 to 3, because that's when they can learn the most information. Whereas on the opposite end, elderly people typically do not have as much plasticity in their brain, which is why they are encouraged to try to keep engaging their brain by doing things such as crossword puzzles or other stimulating activities. So we have two types of synaptic plasticity. We either have um, short or long-term synaptic plasticity. Typically, short-term plasticity, there is a change in synaptic strength for normally less than a second, but it will eventually return back to normal. Whereas long-term plasticity is defined by a long-lasting, activity-dependent change in the synaptic efficacy. So moving into the two types of short-term plasticity, we have first short-term facilitation, and this is done by a rapid increase in the synaptic strength, and it basically occurs when two or more action potentials invade the presynaptic terminal within a few milliseconds of each other. And basically, the short-term facilitation is able to occur because they have an influx of calcium into the axon terminal, which increases the probability of neurotransmitters. So basically, in short-term facilitation, this is all done very rapidly. We have a quick spike. A lot of things happen very quickly, and then what follows is known as a synaptic depression, which is the depletion of the neurotransmitters that were just previously gained, and these depletions are consumed during the synaptic signaling process at the axon terminal. So as you can see, it happens very fast. Um, the CA comes flowing through, and just like that, within a few seconds, there is a depression. That CA2 is consumed, and it is back to its normal phase. Versus for long-term plasticity, we have long-term potentiation, known as LTP, and long-term depression, known as LDT. For long-term potentiation, what we have is a persistent strengthening of synapses based on regular patterns of activity, and these activities basically produce a long-lasting increase in the signal transmission between two neurons. So basically an example of long-term potentiation, since it's based on patterns and it's activity dependent, is if there's a professional athlete, such as a basketball player. He's always dribbling and practicing his game. Over time, long-term potentiation is occurring, and because he is doing this activity dependent thing, there's actually changes that can be occurring in the neurons, which is putting this information and the things that he does into his working memory. And on the opposite end, we have long-term depression. Again, this is long-lasting, and then there's a decrease in synaptic strength. And this is also caused by patterns. So some things that could cause long-term depression are if you have long-term unemployment, long periods of isolation and loneliness, prolonged work strength. And for animals, it could be living in captivity because they are not engaging in all of their natural, instincts, hunting behaviors, etc. So the key things to remember is that in short-term plasticity, it occurs very rapidly, followed by the depression. And in long-term potentiation, it's by repeated activities. This can occur for minutes, seconds, or a lifetime. And on the opposite hand, we have long-term depression, which is also caused by repeated activity on the more negative side, and that can occur for a lifetime as well. Moving on, going more into the different forms of short-term plasticity, we have what is called paired pulse facilitation. Basically, the definition of it is a short-term activity-dependent synaptic plasticity and is most common in chemically transmitting synapses. And basically, it evokes ESPSs. And it happens because paired pulse facilitation happens due to the increase of the presynaptic CA2, and it basically leads to a release of neurotransmitters containing synaptic vesicles. And this is an important part of learning, such as information processing, simple learning, and sound source localization. Moving forward, we have again the opposite, and what you'll realize for plasticity is there's a lot of yin and yang. Normally, when there's a facilitation or a thing that causes an influx, we normally have a depression, which causes things to um, go go a little bit lower than when they were before, kind of as a means to balance things out, such as in ascent potential, when you have that influx of things and then you have that resting slash refractory period that's a little bit lower than the starting point. So in paired pulse depression, basically it acts as the opposite of facilitation and is a decrease in PSPs. And it basically happens because there is a decrease in readily releasable pool of vesicles because they were just used in facilitation. And this can be used as kind of like a way to gain control after a burst of activity. This helps the cells like to have a refractory period, and once that refractory period is over, they are again allowed to continue in their cycle. This is also due to an inactivation of the presynaptic CA2 channels after repeated action from the facilitation process. So moving on, we're going to cover habituation and sensation. Basically, what happened in this study is we had a little a Phyllis Gill, and the researchers decided that they wanted to poke it with a stimulus, probably like a little stick or whatever they had, and they poked it. When they realized that when they poked it, the gill responded. It could have tried to move or retract, or it's just something to basically protect itself. However, they realized maybe for time one, there was a huge reaction when they poked it and it moved and stuff. And maybe by time 10, it moved a little bit less. And over a certain period of time, it barely moved at all. And that response is called habituation, which is the decrease of response strength, basically with repeated times. Moving on to the next image, we can see that more in graph form. As you can see at the top, you see that for trial 1, when it touched the siphon, it went from 0 to 4. So that's a big reaction occurring. That's the first time that the new stimulus was presented, and the animal did not know what was going on, so it had a huge reaction. However, by trial 6, you can see that it is becoming a little more familiar with the stimulus, maybe doesn't see it as much as a threat, and there is a significant drop in the type of reaction. You can see that between 0 to 4. By trial 13, there is basically not really any reaction occurring at all. However, there is a change, and that is because between trial 13 and 14, they decided to shock the tail. And what this did was it made the animal resensitive. It had gotten used to the stimulus, so it was not reacting. However, once they shocked it, it was back up on guard, and it was very reactive to the next thing that touched it, which almost brought it back to um, basically the same reactivity as trial 1. And this is important. Habituation is important. It's natural and it's normal, and it basically allows humans to function in the world. We have a lot of stimuluses around us, and if we were sensitive to every piece of sensory information that happened around us, we would not be able to function in this world. So basically, our mind It tunes out things, the information that is not important, and it creates new memories so we're not repeatedly triggered by the same stimuluses, so that we can move and function in this world without being hypersensitive. However, you can see an example of some individuals that are more hypersensitive would be people that are on the autism spectrum. For them, some things have to be in a pattern, and new senses and experiences can be triggering every time, which would kind of be like a form of sensitization, and it takes them longer to get into that habituation phase. Moving forward, this is the trisynaptic circuit of the hippocampus, and we are currently looking at an image of a rodent. Basically, the trisynaptic circuit is responsible for generating the hippocampal theta waves, and these waves are responsible for the synchronization of the different brain regions, very most specifically in the lymphatic system. And as you can see here, this is kind of like a circuit. Some important players are the pyramidal cells, the scaphoidal collaterals, and these two will be seen later on in the presentation. And basically, there is a circuit flowing, and this is also known as the trisynaptic loop. And it starts here with the prefrontal path, and it continues through, and it hits through the CA3 and the CA1. Moving forward, we've talked about long-term potentiation. However, the rest of the slides are going to cover this in more detail. So just wanted to highlight that long-term potentiation occurs when there is an influx in CA2, and long-term potentiation involves postsynaptic strengthening because of the AMPA receptors. All right, moving forward to the Hebban synapse. A definition of this is the Hebban synapse is a junction between neurons that are strengthened when it successfully fires in the postsynaptic cell. Upon researching this, I found that there are several theories. However, the one that stuck the most is that basically that from how do I say this? There's an increase in synaptic efficacy from repeated and persistent stimulation to the postsynaptic cell. This is also an important key factor of long-term potentiation because it lets us assume that the persistence or repetition of an activity can make long-lasting cellular changes. And this is also associated with the idea called spiked timing dependent plasticity, which is a form of LTP. And the way that spike timing dependent plasticity works is that if a input spike in a neuron happens immediately before the neuron's output spike, then that particular input is made somewhat stronger. However, if the spike occurs immediately after the output spike, then that input is made weaker. And that is why it is called the spike timing dependent plasticity. Okay. And we are moving forward to the LTP of the scaphoidal collateral C1 synapses. So most of what is done with LPT focus between the synaptic connections between the scaphoidal collaterals and the CA1 perimeter cells. And basically these electrical stimulations of the scaphoid generates the EPSPSs. As you can see, normally it takes more than one stimulus to get this function going. As you can see in this graph in A, there is stimulus 1 and there's stimulus 2, and that is what causes the EPSPs to kind of be activated. And typically these cells are, the scaphoidal cells are stimulated 2 to 3 times per minute. And this high frequency can cause long-term potentiation and can increase the length of EPSPs. Okay, moving on, we see the pairing presynaptic and postsynaptic activity causes LTP. And I would like to note that in short-term plasticity, the main thing that is needed is the presynaptic neurons. However, LTP requires a lot of energy and activity, so it needs the help of both the presynaptic and postsynaptic neurons. As you can see here, we have our stimulus that occurs, and with the influx of CA2s, we have a strong polarization paired with EPSPs. So the LTP needs a combination of EPSPs and the cell to be depolarized in order to have long-term potentiation, which shows that just one stimulus alone does not cause long-term potentiation. What we see here in this model is that in order for long-term potentiation to occur, the cell has to be active in order to be strengthened. So as you can see in pathway 1, the cell is active, and the synapse is able to be strengthened with stimulation. And as you can see here, the synapse is not strengthened because the pathway is not active. So even if there's a stimulus or something that occurs, if the pathway is inactive, and this could be because of a type of thing called a silent synapse, nothing is going to happen, which we'll learn more about the silent synapse later. However, as you can see with B, with just 1 week stimulation, it's not able to have the synapse strengthened. However, because there is a strong stimulation occurring on the same neuron, both of these stimulations combined is able to make the synapse stronger overall. We are now moving on to this figure, and this is actually a picture of ionotropic glutamate receptors, and these are basically ligand-gated ion channels which are activated by the neurotransmitter glutamate, and basically they mediate the majority of the excitatory synaptic transmissions throughout the nervous system, and they're key players in synaptic plasticity. So what is occurring here at resting potential, you can see that the Na is allowed to pass through the AMPA receptor. However, on the other side, the NMDA receptor is closed. As you can see, it is blocked by Mg2+, so nothing else is allowed to pass right now between Na. As you can see here in my note, the NMDA receptor is only open during depolarization. So actually what happens during depolarization is that the Mg2 is expelled from the channel, and this allows Na plus to flow through, and it also allows Ca2 plus to come through. And once more of this comes through, the Ca plus, that is what triggers most of our reactions in synaptic plasticity. So moving on here, as you can see, this kind of picks up where we left off. In the last slide, we saw that the NMDA receptor was activated, and there was an increase in calcium concentration. So I'm going to flip back so you guys can see that. You can see the calcium is going through. This NMDA is activated, and you know that because this was the Mg2+ was expelled, and the calcium was allowed to come through. So what happens on this next slide, as you can see, is once the receptor is activated and there is an increase in calcium concentration, which you can see on this side, the calcium is in purple. With an increase of calcium concentration, the CAMK2 is activated. And basically, with this being activated, one or two things can happen. Basically, the AMPA receptors, they can have additional receptors that can be inserted, or what also can happen is that there can be increased AMPA receptor conductance, and that is right here. As you can see, the receptor is right here, and there's yellow spots on it. All right, we're moving on to the mechanism responsible for long-lasting changes in the synaptic transmission during long-term potentiation, and this normally happens during the late phase. And I would like to highlight that the late phase in long-term potentiation can go on forever. It can happen for hours, weeks, or a lifetime. And basically, the late component of this long-term potentiation is the result of PKA, and this is what happens is it activates the transcriptional regulator called the CREB, which in turn turns on the expression of several genes and is able to produce long-lasting changes in the PKA activity and in the overall synaptic structures. Moving on. Here we're going to talk about the synaptic depression in the hippocampus, and we have an image. As you know, long-term depression is caused after long-term potentiation. This is typically due to low stimulation. There's low CA influx, and as we can say, some things that lead to long-term depression could be like low activity, prolonged stress, etc. And so what you can see, what happens here is that we have the cell starting, you can see it's at 100, it's about at its resting phase, it is stimulated, and then after that there is a depression that occurs, and the original point of the cell is at 100. At the end, during long-term synaptic depression, is around 50, and this is kind of like the refractory period, and the cell has significantly decreased after the stimuli was present, and that is due to because the CA2s were mostly used during this height period, and at this point of depression there is not as many CA2s in the neuron. In this final slide, we're going to cover what a silent synapse is. A silent synapse, the definition is it is an excitatory glutamatergic synapse whose postsynaptic membrane contains the NMDA type but no AMPA types. So basically what this means is the silent synapse typically is an immature synapse or a newly formed glutamate synapse, and it does not generate synaptic current. As you can see, it has this NMDA, but it does not have its AMAP, which is needed for long-term potentiation, because that is what allows the CA plus to flow through the cell. So what happens is the silent cells are activated via the insertions of the AMPARs into the postsynaptic membrane, and this phenomenon is called the AMPA receptor trafficking. So as you can see here, we have the silent synapse in A, and it literally has everything besides the last AMPA. So you can see the difference between the silent synapse and the functional synapse. However, once the silent synapse gets its AMPA through the AMPA receptor trafficking, it is now a functional synapse. I'd like to thank you guys for listening to me, and I hope you guys have a great rest of your day.